In this video, we will familiarize ourselves with the apparatus that we will be using in cell and mol laboratory in order to transfer volumes of liquids from one container to another. Traditionally, we use a pipette to do that, but a pipette like this one has a maximum volume capacity of up to 10 ml with volume increments as shown in the graduations of 1/10 of an ml, and that is too large of a volume for cell mol work. So instead, we use micropipettes. Micropipettes can handle volume in the microliter scale. Here we have various types of micropipettes: the P2, P20, P200, P1000, and lastly here we have the P5000 micropipette, which can handle up to 5,000 microliters. Handling the micropipette is very straightforward. So here we have the P5000 micropipette. Here we can see the dial that we can use as a reference for the current volume capacity of our micropipette. And we simply have to grip the micropipette this way with our thumb on the plunger and our index or pointing finger resting comfortably below the micropipette hook. Turning the adjustment knob clockwise or counterclockwise will adjust the volume capacity of our micropipette. We do this by looking at the dial. We have to be careful not to go below or above the volume capacity of the micropipette. Because micropipette tips are separate from the micropipette, we may use freshly sterilized ones in our work. Here we can see it attached to the shaft of the micropipette. Micropipette tips are usually stored inside these containers. When they are sterilized, we usually prevent unnecessary opening of these containers unless we really need to use one. So here's how it would look like. This one is a micropipette tip for the P5000 micropipette. And we have micropipette tips with filters in them. The filters usually protect the micropipette from fluids that we will be handling that are corrosive to the internal mechanism of the micropipette. Notice the different accent colors of the containers of the tips. We use this as a guide in choosing which tip to use for our micropipette. However, sometimes it might be through the actual fitting on a micropipette that we can do this. So here's a tip that would fit our P20 micropipette. We work with the micropipette plunger with our thumb. So we rest our thumb on the plunger, and if we press onto the plunger, we'll notice that there will be some resistance at the first stop. If we press some more with our thumb, the plunger will move on to the second stop. And then we release the plunger slowly as we lift our thumb. We remove the used micropipette tip using the tip ejection plunger, which is located behind the plunger. We press this with our thumb to eject our tip, and we need to make sure that there is a receiving waste basket for the tip.